Hi everyone, it's really my pleasure to be here today and to give you an overview of our clinical infrastructure. So I will cover 3 topics today. Really mainly I'll kind of show you how, a deep knowledge of experience in experimental ASOs and experience in managing trials really supports the professional management of our patients, and how we also are able to then customize the treatment of each patient. I'll then give you a little bit more, evidence of clinical benefit, more than what you saw from Susanna's videos already this morning, but I'll give you some more quantitative data. And then I'll also talk about the safety of our ASOs and provide some information on the safety data we have to date from our trials. So we have a bias to treat patients, right? And that bias is really supported by all the expertise that we have. And that expertise allows us to inform the benefit-risk equation, really, right? So we all realize there is a risk, an inherent risk of not treating a patient with a progressive debilitating disease, right? But we also know, there could be a risk of treating a patient with an investigational ASO, but we know how to mitigate that risk. And again, not to come back to this, but I think 30 years of safety experience that, Ionis has developed and, is still going on in clinical trials today with those ASOs. So that robust experience in ASO discovery and development really kind of supports all the safety experience that we have to date. We are constantly learning from our own trials, right? So we have a few trials ongoing. I'll talk about those later. We're also learning from the commercial trials run by Ionis, by Biogen, by Roche that use, ASOs. And so we learn from those and we bring those learnings to our physicians as well. One thing that's important to understand is, for all our ASOs, we really start with a low dose, right? And I think that's very important. You start low and you go slow. And so you dose escalate in a very stepwise fashion. At every dose, you can go back, look at the results, and then make a decision on what the next dose should be. So it is very truly individualized treatment. And then we have we're creating a safety database for now the trials that are in the clinic. And we have a data safety monitoring board that will be reviewing this data on an ongoing basis. So again, we want to be treating as many patients as we can. But we know that it's very important in order to do this successfully and professionally. So when I talk about successfully here, I mean we need to treat patients with an optimized, individualized ASO that is of good quality, that is safe, and we need to be able to track quantitative and qualitative outcome measures in each individual that is important to that patient, right? So we have to be very focusing on the patient and what is important to the patient. When I say professionally, I really mean more of the how. How do we do this? Well, we do this by partnering with capable and invested physicians and institutions. We are collecting the data in a very standardized manner using a validated system. It's a REDCap system that n-Lorem houses. And we are also, working with the FDA on an ongoing basis. Every time we submit an IND, we get responses. We work with them, very collaborative environment. And so we learn with every IND that we work on. And so really, to do something successfully professionally, you need experience and infrastructure. One thing, side note, I wanted to say here, when we talk about success, we don't necessarily mean, is the ASO working? Is it efficacious? When we talk about success, did we ask the right question? Did we do everything we could to try and answer a scientific question? And if we get an answer to that question, we reach success. But it might not always be that we cause clinical benefit. So I think it's very important to realize that. So at n-Lorem, we have years and years and years of experience. And we're very lucky to be also collaborating with investigators with a lot of experience. One thing that was very important for us is to build a strong clinical infrastructure. We need to minimize the potential risk of ASOs, maximize the opportunity for benefit, and really learn from one patient to the next. So in order to do this, we assembled a very versatile group of leaders with deep clinical experience. And these are leaders that have experience in research, clinical work, manufacturing, FDA interactions, project management, clinical operations, clinical trial oversight. Basically, every function that you have in a big commercial company, we have at n-Lorem. We might not be many people, but we wear many hats, and we have that knowledge. And again, we also obviously partner with subject matter experts that help us find the right treatments for our patients, look at the right outcome measures, and then also, check safety with us. And like Stan mentioned this morning, we really quadrupled the size of the organization in order to meet the demand. So when n-Lorem, started, they realized that they needed to have multiple checkpoints where independent peer review would really support the quality of the work that we do. And so it was very important to reach out to people outside of the organization. And so I'll go through various, a few committees that we have at n-Lorem that are really important, and I'll try to tell you what they all do. So the first committee is the Access to Treatment Committee. So when an application comes in that a physician submitted to n-Lorem, the ATTC will look at the application, look at the specific genetic mutation, determine, is this genetic mutation amenable to an ASO treatment that we could develop. This is really made up of 13 members, and I tried to look at everybody's experience, and I think we are more than 200 years of experience in all the fields that we need: research, drug development, ASO technology, ethics, and patient advocacy. We then have a Study Treatment and Assessment Review Committee. We call it the STAR Committee, and half the time I can't remember what STAR stands for. But I know that that is the committee that advises physicians on which outcome measures to use for their patient. Obviously, the physician treating the patient knows best what measure to use, but the STAR Committee can really sometimes come up with different ideas or different ways of doing things, and so it's always been a really good interaction when we have a STAR meeting. That committee is made of 4 members of a combined 80 years experience in all the fields that are required for that committee. We also partner with experienced investigators who have robust institutional support and staff, and we call those our early adopters, the one that we've worked with to date. And we've learned a lot from them on how to try and do this at their institutions. And we try to take the learnings from our early adopters, and we'll bring that information to the newer physicians that are working with us. And it's been working pretty well so far. And then the last committee that we actually set up pretty recently, now that we actually have patients in the clinic and we need to start tracking clinical data, is our Data Safety and Monitoring Board. And so that committee will review the safety data across all our n-Lorem programs. We might have to expand that committee because once we get all our programs in the clinic, it's basically we're just going to meet on a weekly basis. But they will be looking at the safety and see if there's safety signals across our programs and help us advise on next steps. Now, besides the committees we set up to make sure that there's checkpoints, there's really one big body out there that actually checks all our work every time, and that's the Food and Drug Administration. So the FDA will review, comment on, and approve every IND before any treatment can be given, right? And n-Lorem has received approval across 4 divisions of the FDA: Neurology 1, Neurology 2, these are 2 different divisions, trust me, Cardiology, and Ophthalmology. And we've received positive interactions from all these divisions. And I think this emphasizes 2 things. One, the quality of the IND documents that actually n-Lorem puts forth for the FDA to review. And 2, also the fact that the different divisions at the FDA actually read the guidance documents in a similar way. So they actually interpret them in a similar way, and they approve these N-of-1 trials. Once the FDA approves our INDs, then we have yet another checkpoint, and these are the institutional review boards. So these are the review boards at the various research institutions or hospitals that will review the protocol, the informed consent, and they're really there to protect the rights and welfare of the patients. And like we said, we've worked with many now, and basically every institution has a different IRB, so it's really a lot of people looking at our work. So you might say, well, all these committees, it's going to take forever. But we know prudence and cautious is important. That's why we have the committees. But we also know that time is of essence. And so we try to do things in parallel to be able to move faster to get to a treatment for our patients. So I'll run through you, to show you what that means. So this is really kind of like the workflow. If you look at just like the research and GLP side of things, so at that first star on the left, I hope it's the left for you too, I get confused. That's when an ATTC, when an application comes in, then we need whole genome sequencing and patient cells for us to be able to start ASO discovery. So once we have that, ASO discovery starts, and Julie did a great job at telling you everything that's involved there. That star after the ASO discovery is when we decide on an ASO lead. We put it into GLP tox, manufacturing, and then we can submit the papers for patient treatment. So that's one part of the work that's going on at n-Lorem. But in parallel, there's all these clinical activities that have to happen. So pretty much as soon as a program moves into ASO discovery, we start working on treatment goals with the physicians and prepare for them to present at the STAR meeting. So once we have finalized the treatment goals, we can then go on and write the protocol and finalize that individualized protocol and informed consent. Once we have a protocol that's done, we can then go on and develop an electronic clinical database that will be able to capture all the pre-treatment clinical data for the patients. And then again, in parallel to all that work, we also work on the IND documents and on the submission process. We submit to the FDA. Once the FDA approves it, we submit to the IRB. Once the IRB approves it, then we can start collecting, or we can start treat the patient and collecting all the clinical data post-treatment. And so here, the different colored stars show you where all the committees fall. So the ATTC committee is obviously at the very front. The STAR committee will review the treatment goals. External roadblocks, FDA and IRB will be the ones giving us that final okay. And then the DSMB will review the data once the patients are in the clinic. So we've set up this infrastructure. We are very, not optimistic. We know that this infrastructure is scalable and that we can support, the treatment of many more patients to come. We know that because we've, institutionalized standardized protocol language, template informed consent language. We have a streamlined process to support IND filing. It's still a ton of work, but we know the steps to do it and to do it quickly. We've also had really good interactions with the FDA and responding to FDA queries. Again, I think what is really important is our electronic database. It's a REDCap database, an academic one that a lot of our investigators have used for their own clinical trials. We've been able to standardize, some of the forms that we use. We've developed a large library of electronic case report forms that we can use from one program to the other program. And because, for example, for a neuroexam, we've made sure that everything that we think is important is on that neuroexam case report form, and we can use it for one program and another program and another program. And so we can actually, easily afterwards compare the data from one program to the next. And I think that's really a strength of how we've set that entire infrastructure up. So let me give you a little bit of where we are today. And Stan kind of gave it away this morning, but it's okay. We've submitted 9 INDs in the last 13 months to 4 different divisions. Most of the INDs to date, so 7 of them have gone to the 2 neuro divisions, 1 to the cardiology division, and 1 to the ophthalmology division. n-Lorem pretty much authors 98% of that content. We work with our investigators really to describe the patient, because there's a section in there where we describe every patient and their clinical status and why they should get this N-of-1 medication. We've had more than 25 FDA interactions. This is between meetings with the FDA and responses with the FDA, and they've all been very positive. And we are expecting to submit 2 more INDs before the end of the year. So once we're done here, we all better rush back to California, wherever we're from. But yeah, so we're expecting to do 2 more, at least this year. In terms of clinical metrics, 5 patients have been dosed to date, 4 CNS patients, 1 eye patient. If you do the math, you're wondering what's up with the other 4. We still have 2 INDs that are under FDA review, 1 IND is under IRB review, and 1 IND just received IRB review, and the site is planning, is setting up for a visit for the patient. So that gets you to 9. Patients have received between 1 and 6 doses, and I'll show you a table later to tell you what that means. And to date, I think what Stan also mentioned earlier, we have not had any ASO-related adverse event or serious adverse event, which is fantastic. So now let me change gears a little bit. Now I just want to show you or give you an example of how we really customize the needs for each of our patients. So like Stan mentioned this morning, right, 2 patients can have a mutation in the same gene, and it can lead to very different phenotypes. And so it's very important for us when we work on the treatment goals that we really find treatment goals that are important to a specific patient, right? So I'm giving you here an example of 2 patients. that we have in our pipeline. One has started treatment, one is about to start treatment. Both of them has different mutations in SCN2A gene. So one is more of a gain of function, the other one is a mixed gain of function, loss of function. However, there are some symptoms that are similar across both patients. And so we'll track seizures for both patients via seizure diary. We'll also track behavior and communication and development in both patients. But then both patients also have different symptoms that we'll track. And so for one patient, it's really focusing more on the behavior and communication. So we have added measures in that realm. And then for the other patient, it's more motor issues and gastrointestinal issues. And so we'll have scales that focus on those symptoms. So this is really just very high level, obviously, but just trying to show you how we try and individualize treatments for the different patients. Stan, obviously, this morning already showed you great videos from Anna and Susanna. And really, I think, especially Anna, led us to understand that even if you start treatment late, you can still have a benefit, or what some people would consider late. And so it's never too late to try something. What I'm going to try and do here is give you a little bit more quantitative data. You've all seen videos of Susanna, so I think everybody has drank the Kool-Aid, hopefully, on that treatment is working for Susanna. But I'm going to try and give you quantitative data. One thing I want to mention here, because we are really normally blinded to every application that comes in. So we do not know the name of the patients that come in unless the family identifies themselves to us. So obviously the Rosen family has identified themselves, but Susanna is also in our database tracked by a number, which is 255. So just wanted to make that clear. Some patients we know, some patients we don't know. So she has a mutation in KIF1A gene, and as Stan said, this encodes for microtubule motor protein that's responsible for transport along axons. When Susanna started treatment, she was 8 years old, this was just a year ago, had severe epilepsy, intellectual disability, and very, very painful and debilitating neuropathy. She also had to start using a wheelchair because she became non-mobile. This is the protocol that was written, or timeline protocol that was written for Susanna specifically. So a couple of things here I want you to note. First of all, it's a 12-month study, right? She's where the big red star is. She's received 6 doses to date. The doses are also mentioned in the graph. So you see we go, like I say, we start at 20, we go slow, and we increase and kind of wait to see what happens and go to the next dose. You can see all the assessments that are done. So a lot of it is for safety, vitals, EKG, overnight EEG, but then also some, to measure how she's walking, 6-minute walk test, and so forth. The one thing I want you to realize is that little red star in the middle. These are assessments that track gross motor function and fine motor function, or fine motor skills. And these were actually added in the middle of the trial because these were things that she now was able to do. And we didn't really know to track those at the beginning because she wasn't able to really do some of those assessments before. So I think this is also something that we learn, and it's fine to add that later on and to start tracking it later on. All right, so quantitative data here. So just to orient you, it says parent-reported seizure data on the top, falls in the middle, and quality of life on the bottom. If you look at the y-axis, that between the first 2 red vertical lines, that's the pre-treatment time. So that's our baseline, pre-treatment baseline. Every vertical line thereafter is a dose, right? So she had a ton of seizures before treatment. And over time, and really actually pretty quickly, the seizures started to come down, right? And so she still has some seizures, but it's definitely much reduced from what it was before. In terms of the falls, I think you can say, see the same, but I think what's important to note here, remember she was really using a wheelchair a lot at the beginning, so she was actually not walking that much and would then fall because of the disease. But now, even though the falls have decreased, she's actually walking a lot more than she did at the beginning. So I think this is really something to kind of point out, and something that one needs to understand if you also understand what the child is actually doing during their day. And then the last one is, quality of life that's increasing. You always want to see that increase over time. And then finally, I think what is really important is that there's no ASO-related adverse events in Susanna. And then additional things, again, you've seen the video this morning, so I feel this slide is a little bit redundant here, but additional comments that we got from the families on things we were not necessarily measuring in the trial, but things for us to think about whether we should include it in future trials. Her tremor is almost all gone. She can hold a baseball. She made a bracelet for me today, so I'm very happy about that. She can sing, something she hadn't done for a while. And what we heard is also the pain has really gotten much better in her hands and her feet, but the pain does come back towards the end of the dosing interval. So again, this is something that with the physician, the family will have to see and try and fine tune and maybe increase the dose or reduce the dosing interval. So all things that we'll have to think about, think through. And she's able to make it through the day without a nap, which I think that really talks about the stamina, which is very important. The 6-minute walk test supposedly tests a little bit stamina, but not really. It's also, in the clinic. So it's really nice to kind of hear from the parents on that. So another patient, and I'm running over, but that's fine because I think this is very important. Just wanted to give you some baseline or some data on this patient, an SCN2A patient. So SCN2A is a voltage-gated sodium channel, and the mutation leads to hyperexcitability of neurons due to an increase of sodium flux. So when the patient started dosing, was 9 years old, had severe intractable epilepsy, that was uncontrolled by anticonvulsants, had to be on really high levels of phenytoin, cognitive language and adaptive language adaptive impairments, and nonverbal. So for this patient, actually, you will see that the protocol is slightly different. This is a 24-month study now, so 2-year study. And mainly, this is due to the fact that if you want to track neurodevelopmental, cognitive, language, adaptive behavior endpoints, you will need more time than just 12 months. It's very unrealistic to think you're going to change, everything in 12 months. So this patient has received 3 doses of the drug to date. And so very preliminary data. I'm happy to share it with you, but I want all of us to think this is very preliminary data. Again, orienting you here, number of seizures on the y-axis and months pre and post-treatment on the x-axis. So the first 3 bars represent the 3 months before treatment. And you see, you don't need to know the number. You can see the height of the bars. And then treatment started at 20 milligrams and 40 milligrams. And you can see that the seizures were coming down from then. So we'll have to track that. Obviously, there's other medications being given. And I think, we always try to keep the other medications constant. But sometimes you also have to just take care of the patient and do what is needed at the time. So now moving on to safety quickly. Really, the bottom line is there's no ASO-related adverse event or serious adverse event that we've seen to date. The table below is showing you the number of doses that each of our patients in the clinic have received and what the highest dose level was. First 4 are intrathecal injections. The last one is our intravitreal patient who just got dosed a couple of months ago. One thing to note is the first 2 patients got 3 doses. They were very advanced when the study started and unfortunately progressed further, making it impossible for them to come back to the clinical site. So technically, treatment has stopped for those patients, but the ASO had no side effects. But in those patients, we probably were a bit too late to be able to, have an effect. One thing I just wanted to also mention is, like we said before, the work that, Stan did at Ionis really, helped us in terms of all the decision making that we're doing here in the clinic. There's a huge number of papers that they published, integrated safety databases that they published, from non-human primate toxicology studies to controlled clinical trials, both with drugs given systemically and then also with ASOs of the same chemical class and the ones that we're using, when given intrathecally. So there's really a ton of information out there in terms of the potency, safety of these ASOs. And the papers that are listed here are obviously the nusinersen papers, the papers for the AD drug, Huntington's drug, SOD1 ALS. So, there's really a wealth of information that we all have, and especially some of us, came from Ionis, others didn't, but, definitely the technology and the platform, is not new, and we know the safety background. So lastly, before I finish, I just wanted to, say 2 things. It's very important for us to share data. I mean, we wouldn't have organized this meeting here if we weren't about sharing data. And we think it's important to share with the medical and the patient community because, knowledge that we gain when we give it out to you guys. That's how we will really make progress in the future. So we want to do this in presentation. We think it's very important to do this in peer-reviewed journals, case studies that will be presented, and so forth. The way that we set up a REDCap system, like I said, it's because some of the forms are very standardized. It's going to be easy for us to pool data across our programs and to be able to compare different data from the programs. Let's say, do seizures, are seizures affected similarly with different ASOs, if the mutation is different. Like, we can easily look at that because our systems allow us for that. Integrating our clinical data sets with other sources is certainly always a discussion, but I think it's very important to realize we have to make sure that the data that would be combined is of quality and has followed a rigorous process of collection. And I think for me, it's very important to also make sure that we protect our patient information and that and the use of the data. And so we need to make sure that if data is pooled, everything is done in an ethical manner, and everybody knows, especially the patients, know how the data is used. So with that, I hope I have shown you that n-Lorem has successfully industrialized the development of optimized ASOs. We have a clinical infrastructure that is solid and that is delivering value to our patients. And we are ready to meet the needs of tomorrow's patients. We know we can do it. We're very secure about that. We appreciate the FDA's collaboration. We've successfully navigated the regulatory landscape across 4 divisions. We're ready to take on the other divisions. n-Lorem is sharing clinical data, and we want our investigators to publish and to present the work that we do, and we're treating patients. So we're very honored by all the families who've put their trust in our hands. And with that, I want to thank every institution that has submitted an AS or a patient for us to consider, and also thank the collaborators, and pretty much thank everybody who's on the same page than us and who aims to advance the treatment of nano-rare. So thank you very much.